Hi, and welcome to Pharmacology of the Synapse. In this video, we will explore the function of the different nicotinic receptor subtypes using drugs. In this segment, we will briefly go over some nicotinic receptor agonists and antagonists, and the pros and cons of using pharmacology to determine receptor subtype function, as well as some examples of how pharmacology has been used to identify a role for the nicotinic receptors in drug addiction and cognition. Many of the agonists and antagonists that act on nicotinic acetylcholine receptors are compounds derived from plants or animals which are used as protective or offensive mechanisms in nature. The endogenous neurotransmitter for the nicotinic receptors is acetylcholine. Some of the agonists that are used in research include nicotine, a compound from the tobacco plant, which the receptors were named after, cytosine from the golden chain tree, and epibadidine from a poison arrow frog. These compounds are generated in these plants and animals as a deterrent from predators. Many antagonists for research were also generated from nature and include DH-beta-E, a compound derived from coral tree flowers, alpha-bungarotoxin derived from a poisonous snake, and alpha-conotoxin, which is derived from a poisonous snail. Like the agonists in the previous slide, these compounds are generated as offensive or protective measures. A synthetic antagonist that has been widely used in research is mecamylamine, which is also called Inversine. This drug has actually been used in people and was prescribed as an antihypertensive long ago, but has since been taken off the market. If we want to figure out what behaviors a particular nicotinic receptor subtype contributes to in vivo, we can use agonists and antagonists that affect the subtype and observe the effects on behavior. The pros of using pharmacology are that many different types of drugs are available that can be used. Second, the experiments are relatively straightforward and usually involve administering the drug to an animal and observing their behavior. We can also use pharmacological principles to verify that a certain subtype is involved. For example, an effect observed with an agonist should be blocked by pretreatment with an antagonist against that same receptor subtype. The cons involved with using pharmacology to determine receptor function include the fact that many drugs hit multiple receptor subtypes. Nicotine, cytosine, and mecamylamine are nonspecific and affect all receptor subtypes. Epibadidine primarily binds to the alpha-4 beta-2 subtype, but can also hit other subtypes. Erythroidine binds to receptors that have the beta-2 subunit, but this could include many different subtypes. Alpha-conotoxin and alpha-bungarotoxin are slightly more specific. Alpha-conotoxin binds to alpha-3 and alpha-6 containing subtypes, which again could still be quite a few, and alpha-bungarotoxin binds primarily to alpha-7 only receptors. Here is an example of how pharmacology can be used to narrow down the receptor subtype that is responsible for a particular physiological effect. Sensitization to drugs of abuse is a process by which repeated drug administration causes an enhanced response to the drug. This process involves changes in neurotransmitters and neuronal function, and is associated with other addiction-related behaviors. Locomotor sensitization to repeated nicotine administration is often investigated in animals as a form of drug sensitization. So, which nicotinic receptor subtypes are involved in nicotine-induced locomotor sensitization? Nicotine itself activates all nicotinic receptor subtypes. In this study, repeated injections of nicotine in the black bars here results in greater locomotor activity compared with a single acute injection of nicotine in the hatched bar. Injection of mecamylamine attenuates the locomotor sensitization seen in the gray bar. However, an injection of alpha-bungarotoxin does not alter nicotine-induced locomotor sensitization. This suggests that alpha-7 receptors are not involved in this process. But since mecamylamine is a nonspecific antagonist, the receptors involved in this process cannot be identified using mecamylamine. Which nicotinic receptor subtype is involved in cognition? In this experiment, a radial arm maze task is used to investigate working memory. In this task, rats or mice are trained to enter the arms of the maze for a reward. If the animal goes into an arm that it has already visited, this is considered an error in working memory. The number of different arms that the animal enters before it makes a repeated entry is a measure of working memory. In these panels, you can see that the nicotinic receptor antagonists mecamylamine and DH-beta-E cause the animals to re-enter the arms that it has already visited sooner compared with no drug administration. This suggests that nicotinic receptors in the ventral hippocampus are involved in working memory because these antagonists were infused into this particular brain region. The use of DH-beta-E, an antagonist that affects beta-2 containing receptors, suggests that beta-2 containing receptors are involved in working memory, but this would still implicate many possible combinations.